Cellular respiration begins with glycolysis, when one glucose molecule has 2 ATP added to produce 2 pyruvate. Each pyruvate then binds to an NAD, and the NAD strips an electron and turns into NADH. The NADH from each goes into oxidative phosphorylation to be used later in cellular respiration. Then, the pyruvate is turned into acetyl-CoA, still outside of the mitochondria. The acetyl-CoA then goes into the citric acid cycle. When acetyl-CoA is produced, a molecule of carbon dioxide is also produced by the cell. Acetyl-CoA enters the citric acid cycle in the inner mitochondrial complex. The CoA breaks off from the acetyl-CoA, forming another carbon dioxide. As the 2-carbon chain enters the Krebs cycle, NAD and FAD come in and strip electrons off to become NADH and FADH2, which then both move into the oxidative phosphorylation to be used later. More carbon dioxide is produced as a result of the 2-carbon chain moving through the Krebs cycle. Another 2 ATP is also produced. The FADH2 and the NADH that arrive in oxidative phosphorylation are reduced, and NAD plus and FAD plus go back outside to be reused later in other cycles. Then the electrons that come off enter the electron transport chain, where electrons move down each protein, going to a lower activation energy. This allows H plus ions to be pumped across the membrane to be on the outside. It also powers the ATP synthase at the end, in which H plus ions are moved through it, causing the bottom of the ATP synthase to spin, which then bonds a phosphate group to an ADP, producing ATP.
And a total of 30 to 32 ATP are produced from this.
The electron exiting the 4th protein will bind to an oxygen waiting at the bottom of the staircase. This will make it more susceptible to bond to hydrogen, and it will form H2O as a byproduct of this system.